Hello everyone, welcome to Get My Class. So let's solve this question here. It says DNA fingerprinting involves identifying differences in some specific regions in DNA sequence known as dash. Option A, whole genomic DNA. Option B, repetitive DNA. Option C, non-repetitive DNA. And option D, cDNA. Now let us understand the question. It says DNA fingerprinting involves identifying differences in some specific region in DNA sequence. So what is that known as? Okay. Now before we look into the answer, first of all, we have to understand what is DNA fingerprinting. DNA fingerprinting. DNA fingerprinting, as the name indicates, it is a procedure to identify the blueprint, or I can say it gives the details of the blueprint of the genomic DNA of the genomic DNA. Okay. Now, when I'm saying blueprint, that means it is talking about the specific sequence. So, in DNA fingerprinting, we try to identify specific sequence of DNA. Specific sequence. And which sequence are we talking about? There are specific sequence which are called as the repetitive DNA. Repetitive DNA. Repetitive DNA is nothing but that part of the DNA where you can see that certain sequences are repeated, again and again. And this repetitive DNA, the sequence of the repetitive DNA, the size of the repetitive DNA will vary. Okay, so this is specific. The repetitive DNA is specific for an individual. Okay, for in every individual person, this repetitive DNA is specific. Okay, we all know that there are about 99.9% of the genome which is similar, right? All the human being, if you see, every human being, if you compare two people, you can see that 99.9% the sequence and the genome will be similar, but the 0.1% will be different, and that 0.1% will include this repetitive DNA. So it is specific for an individual. In terms of what? In terms of length of this repetitive sequence, in terms of the sequence itself, what type of sequence is present. So that also will vary. Okay. So by knowing the length and the type of sequence present in the repetitive sequence, okay. So these, as I said, these all contain the repetitive stretch of the DNA, and the sequence of repetitive stretch will vary from person to person. Even the length will vary from person to person. So let us see the answer. Option A says whole genomic DNA. No, and it is not even possible, to consider the whole genomic DNA, because they are supposed to be running it on the gel, and further they are going to look for the band. Okay, and this will whole genomic DNA. It is not considered in case of DNA fingerprinting. It is only the important part that is called as the repetitive DNA. Option B says repetitive DNA. That is absolutely right. And this repetitive DNA, during centrifugation, it gets separated from the main DNA, and it forms a separate band, and that we call it as a satellite DNA. Okay, satellite. Okay, so satellite DNA, why? Because it is getting separated from the rest of the genomic DNA. That's why we call it as satellite. Now again, within this repetitive sequence, you have again tiny, smaller part of repetitive sequence, which we call it as mini satellites, micro satellites. You have different varieties here, micro, mini, etc. Okay, and these are very helpful in forensic science. Forensic science. Okay, so as to look for the criminal, and to collect the sample from the place where the this one has done, or criminal act has been conducted, and then they will compare that particular DNA sample with the suspect's DNA sample. Okay, so option B is the correct answer. Option D says non-repetitive DNA, which is incorrect. Option D says cDNA. cDNA is nothing but the complementary DNA. So here we are not looking for complementary DNA. We are not even going to synthesize any complementary DNA. So the correct answer is what? The correct answer is option B. So let us conclude. Which is the correct answer? Option B is the correct answer. I hope you understood the explanation. Thank you, everyone.